Good evening, everyone. Am I audible?
Yes, ma'am. Good evening.
Yes, ma'am.
First of all, Happy New Year to all of you. Hope you all are doing good. This is 2021 right now, and this is the year you will be appearing your exams. So be sincere and be serious about your studies. Okay. So for today, first of all, we'll be discussing a little bit about economic botany. After that, we'll be going for the rest section we left in the last class for apomixis. Then we'll be seeing the new chapter, that is your self incompatibility. Okay, so last class I told you regarding some history in the economic botany section, that is the famous authors and their books and all the names. They are also very important. So today we'll be talking about the flower. Okay, flower. Why flower? Because flower is related to the flower is the basic part which is involved in reproduction of plant. Okay, so let's see the typical flower structure. So this is the structure of a typical flower, as I told you in the last class. Here you can see this from outside to inside, the sepals, then petals, then your stamens, then carpel is there. Okay, this is the female reproductive part. Here the pollen will land and it will go, and this is the ovary. Okay, inside ovary you can find the ovule, and this is the holding position, that is your receptacle. Then both stigma and style, they will constitute stigma, style and ovary, they will constitute the carpel, that is the female reproductive organ. And the anther and filament, they will together constitute the stamen, that is the male reproductive organ. Okay, so I already told you all the structures and what their functions are. Okay, and I also megasporogenesis. Now there are some important one-liner questions which I want to discuss with you. Like, you have to answer, okay, based on what you know and what you don't know. I'll be explaining the rest part. Okay, then tell me, uh, ensembles, group of stamens called as, please answer. Group of stamens, what we call that as?
Androsium.
Voice not audible right now. Hello? Hello?
Ma'am, voice is not clear, ma'am. So ma'am, we are getting confused.
Is it audible now? Hello? Anyone can tell whether it is
Yes,
audible or not?
yes, ma'am. Yes, ma'am.
Okay. So what I asked you is, group of stamens will be called as, a term is there. What is that called? Group of stamen?
Ma'am, anthers. Ma'am, androsium. Androsium.
Yes, group of stamen is called as androecium. Likewise, group of carpel will be called as gynoecium. Okay.
Gynosium,
Then group of sepals, yeah, group of sepals will be called as
Calyx. Calyx.
calyx,
calyx. Group of petals will be called as corolla. Okay.
corolla,
And as I already, yeah, as I already told you, the flowers which is lacking any of these four parts will be called as incomplete flower. Okay. Then the flower which is lacking the corolla only, that will be called as, there is a term for that, that will be called as corolla section, otherwise called as petals, right? So flower which is not
petalus,
having corolla, it is called as apetalous
epipetalus,
flower. Okay, that is not epipetalous, that is
um,
apetalous.
epipetalus,
Yeah, that is, let me write it down here. That is called as apetalous. Petalous. Okay, means A means absence. A means absence. Petalous means petal is absent in the flower. The example of such a case is, you can see in case of annona. Annona means, that is a fruit. The common name is, what do you call that? Sitafal, that is called. That is
Sarifa,
custard apple.
custard apple, Sarifa,
Yeah. So in case of annona, you can see this sort of apetalous structure. Okay. Then the flower which is having both stamen and pistil together, what that flower will be called as? Yes, means both male and female reproductive part in same flower. That flower will be
Hermaphrodite.
called as,
Perfect.
hermaphrodite,
Perfect.
yes, yes, that is called as hermaphrodite. Okay. Then, okay, the means the study of, not the study, means the observation of position of calyx corolla in a flower bud, means how they are positioned. That thing is called at what? That arrangement of flowering parts in a flower is called as, there is a term for
inflorescence,
that, elevation. Okay. Aestivation. Estivation. Okay, so based on which type of floral arrangement you can see, so on that basis, all the flowers have a particular type of estivation. What is inflorescence, are you asking? Is a structure, or it is a but in a complete
no ma'am,
means, if this is the inflorescence, yeah, yes, what you asked.
no, I didn't ask anything, it was just,
Okay,
I
okay, you
thought,
just said
yeah,
the answer. Okay,
no,
so that,
actually,
Yeah, please pick up clearly. I don't understand what you said.
Ma'am, your voice is not clear, so I thought you were asking arrangement of flower on branches.
Yeah,
I think you were.
no, no. I asked like arrangement of flowering parts of flower in the flower. I asked regarding that. Okay. Then the next thing is study of pollen. There is a term for study of pollen. What is that called? Yes. Anyone? No. Study of pollen is called as
Palynology.
palynology. Okay. Palynology. That is study of pollen. Okay. Then palynology, that is study of right now.
Yes.
No. Okay. So basically, depending on which family the particular plant belongs to, the flower will have a particular kind of structures, like their stamen arrangement, their petal arrangement, their sometimes what will happen is stamens are fused with petals, stamens are fused with anthers, so they will be fused at top, they will be loose at the bottom. Based on that, there are several terms mentioned. Okay, so based on Ramya just okay, so if the stamens means in the flower, in this sort of flower, if all the stamens are attached to all together, means they form a column kind of thing, means it is the stamen. It anthers will be on the top, anthers like this, and the stamen sort of condition will be called as monadelphous. Okay, monadelphous condition where all the stamens means all the filaments are connected together. Only the anthers will be free, little bit on the top. That
Cereals.
condition is called as monadelphous. Yes.
Cereals are its example.
No, cereals are not its example. Its example I can find in it now. This condition in case of Malvaceae family. Can anyone tell me any plant which belongs to Malvaceae?
Cotton,
Okay. Okay.
cot and okra.
No, from this section actually, yeah, cotton. Cotton is one example. Okra is another example. Yes, means those two
Okay.
are famous examples. Apart from that, also many examples are there. But cotton and okra are famous examples of Malvaceae family. I'm sorry, I'm having a little bit cold. That's why. Please excuse me for that. Then stamen attached to form the two groups of filament. Means the condition
Didelphus.
is like where this stamens are like this, and they will be attached in, yeah, that will be called as means two columns will be there. And somehow means which arrangement that will be there might be a condition that if totally 10 and 10 stamens are there means 9 plus 2 9 plus 1 this sort of
9 plus
arrangement
1.
might be there. Means 9 will be grouped together, one will be and means kept aside, or 8 will be grouped together, 2 will be kept aside. That condition is called as didelphous condition. Didelphous condition, where you can find it.
Leguminous crops.
You can find this condition in case of your Fabacy. I just for your understanding, I'm explaining the condition. What you need to remember is the condition and which family it is found. Okay, like monadelphous condition is found in case of Malvaceae. Diadelphous condition is found in case of a Fabaceae. That is all the legumes. Fabaceae is otherwise called as Leguminosae. Yeah, legumes. All the legumes you can find this. Okay. Then if the condition is two or more groups will be there, means not two, more than two groups will be there, that condition is called as polyadelphous. Okay, polyadelphous condition you can find in case of your castor. Okay, now can anyone tell me what is the family of castor? Yes, I asked you like what is the family of castor? Anyone? So castor belongs to your Euphobiaceae family. There is a characteristic feature of Euphobiaceae family is that if you cut the plant of Euphobiaceae, means if you just bend a stem of Euphobiaceae family any plant, you can see that white color fluid will come out. So mostly whichever plant is on breaking, you can find that sort of white color fluid coming out. They all belong to Euphobiaceae family. So castor belongs to family Euphobiaceae. And scientific name of castor, anyone?
Ricinus
Yes.
communis.
Resin is communal.
Yes. And there is another important chemical associated with castor, which is a toxin. What is that? It is resin. Okay, resinous communis. Most of the chemicals know from the scientific name only. Means if even if you don't know, just for guessing purpose, I'm telling you, most of the chemicals, no, they are taken on the basis of the scientific name, because it is taken from the resinous communis. So the scientific name means the chemical's name is resin. Okay, so like that only sometimes you can guess also like what will be the chemical component. So this is a toxin actually. So castor is not edible because of this particular chemical. Okay, then what else? Okay, there is a process in physiology where low temperature exposure will induce the floral induction, means flowering structure formation. What is the process is called?
Vernalization. Vernalization. Vernal.
So I was telling you about the process of vernalization. So vernalization is a process where you expose the plant to low temperature, and that particular process will lead to alteration in the internal hormonal label of the plant, and ultimately it will lead to the process of formation of your means formation of your flowers. Okay, it is raining over here, students, so please excuse me for bad internet. What to do? Then okay, if there are three conditions you can find in case of your ovary. If the ovary position is a epigynous, hypogynous, and perigynous, three conditions are there. Epigynous means, anyone can tell me what is epigynous
Ma'am,
conditions?
ovary is situated above other anther parts. Ma'am, it means that attachment is above all other parts.
What about hypogynous then?
It is lower to other parts. That means ma'am, its attachment is lower to other parts. And perigynous, ma'am, I think equal.
So what happens in case of your epigynous flower is your epigynous or hypogynous, all the conditions are basically we used to tell that based on the position of where the particular part is present. Okay, the we call the flower as hypogynous if sepal, petal, and stamen everything is below ovary. If it is present below ovary, then the condition is called as hypogynous. If the parts of the flower are above ovary, that condition is called as epigynous. And likewise for perigynous, what they will say, just give me a second. So perigynous condition is there where almost means will be called as your perigynous. Okay. Then coming to the next thing regarding this particular aspect, that is Okay, that was about your ovary position. Okay, so inside your ovary you can see particular kind of septums are present, means partition walls are present, and in the
Mm.
ovary you can find that ovules are arranged in a particular way. Okay, so the way ovules are arranged on the ovary, that particular condition will be called as placentation. Okay, means after the means if it is the ovary, let me take the pen. If it is the ovary, inside ovary you can find that in this particular condition, this is the single ovule I have showed you. If the condition will be like this, and ovaries are arranged either in this linear way like this, or they are arranged in a circular way, means here one means circular arrangement is there in the ovule, or what sort of arrangement ovule is having inside the ovary, that thing is called as placentation. Okay, so based on that we can find various types of placentation. Then so this is a little bit about your flower, bit regarding type of fruits. Okay, so fruits will be based on, means basically there is a classification of the fruits. Sometimes they used to ask like which type of fruit the particular plant is. Like say for mango there is a type of fruit is called as, which type of
Droop,
fruit? mango
droop.
is, mango is drupe. Yes, there is another type of fruit is called berry. Any example you know from berry?
Yeah. Banana.
Banana is a berry, tomato is a berry, and there is one peculiar example, that is erica nut. Erica nut is a nut, but it is the fruit type is berry. Okay, so remember that sort of exceptional points. And there are some fruits which are called as pseudocarpic fruits, means they are not really, means the fruit is not from the original ovary. Okay, and the example is some sort of
Apple.
Apple.
apple. Yes,
Thalamus.
the, you can see there that the origin of the particular fruit is not from the ovary. Here the thalamus has
Thalamus.
enlarged to form the whole fruit. Okay, then there is a group of fruit which is actually aggregate, means aggregate of many ovaries together.
Custard
uh,
apple. Jackfruit.
yeah, custard apple, jackfruit, those you can find, and there is
Fig.
several specific names for them. If possible I'll try searching the names, and I'll try to
Itario of berry.
Itario
Itario.
berries. Yes, possibilities Itario berries. I'll try searching the list of it and give you. Right now I don't have it. I'll give you. Okay. Then for the particular crops like crop plants, which you'll be studying like rice, wheat, maize, and all those things, which particular fruit, means which particular type of fruit the particular plant
Caryopsis.
carries, that thing you need to remember. For cereals it is mostly caryopsis. Yeah, what is the
Caryopsis.
peculiarity of caryopsis?
Lemma and Pelia.
Lemapelia is found, but the caryopsis, that type of fruit has the peculiarity that the seed coat is fused. Fused seed coats. Seed coats are called testa and tegmen. Okay, if this is the seed, if this is the particular seed, the outer coat, outer side coat is called as testa, and now the inner one called as tegmen. When
Take.
both the and inside you can find your particular what seed. Okay,
Yes.
and this is the total fruit. When everything is fused, that particular condition is called as your caryopsis. Okay, and it is a means ovary is totally united with the seed coat. Okay, so that is a little bit regarding your economy botany section. Let's move on to apomixis where we left. So I think in the last class we studied all the means type of apomixis, how the apomixis is getting conducted, and we stopped here before genetics of apomixis. Okay, isn't it? Or did we move further? I guess here we stopped, right?
Yes, yes, ma'am.
Yes, ma'am.
Okay. Okay, so I'll begin from here itself. So here I discussed like in genetics of apomixis, the process is governed by one or more than one gene. So basically, in most of the cases, it is governed by one or two genes, and if it is governed by one or two genes, it is means it is major gene and it will have either dominant or recessive in nature. But sometimes it is polygenic also. In some cases, you can find, in case of your pennisetum and panicum and citrus and trypsacum, these are famous examples where you can find the single gene locus governing the trait. That too, in case of your panicum and citrus, you can find the dominant gene action directly. Okay. And recently, like polygenic nature is now recently only discovered, and this point is very important, that in Pennisetum ciliaris and Cenchrus ciliaris, it shows the obligate apomixis, that is inhibitory gene interaction is found there, and because of that it shows the obligate gene action. Okay, that also I told you regarding this. This is why this is the famous example of Herasium, that is apomictic in nature. Where else you have studied the apomictic nature of Herasium? Anyone?
Ma'am, Mendel failed to prove his laws on Harrison because of
Yeah,
apomixis.
Heracem is otherwise called as hawkweed, and he this is the plant where Mendel failed to
Failed.
demonstrate his laws. Why? Because this is not normally means normal sexual reproduction is not there in particular plant. Here apomixis is present. That's why he failed to explain this. Okay, so here you can see what I am trying to show you here. Here the normal megaspore mother cell is there in your ovule, and in that ovule the normal gametophytic program is occurring, means from this it is going for gametophytic program, and further here you can see four cells are there. These three will degenerate and this one will remain, the upper one will remain, and this is going further for the differentiation to form your embryo sac, and for the your double fertilization event is occurring, and after that formation of seed is there. Okay, but in case of your apomixis in the lower section, as you can see, from that particular megaspore from this particular cell, which is the megaspore mother cell, from here the division is somewhat aborted. Okay, so from here directly it will go for the so from this particular cell will go for the formation of embryo sac directly, without undergoing the particular sort of division which was destined here. Okay, so you can find this difference in the particular seed over here and over here. Here there is no double fertilization event like it is occurring in the normal case. Okay, so here the seed development is fertilization independent process, right? So two developmental changes that occurs in apomixis than in normal is a suitable modification or failure of meiosis during megaspore genesis. As I have showed you, here there is no, here there is meiotic event is going on, here the meiosis is either completely absent or modified. And second thing is that there will be initiation of mitosis in unfertilized 2n egg cells and some other 2n cells of the embryo sac. Here what is happening is this particular cell, this particular megaspore mother cell has the ability to undergo the program to form the seed. None of the other cell can undergo that particular process. But in case of apomixis, you can find that abnormality, that all the 2n cells over here from any of this, depending on the plant type, from any of these particular 2n cells, it may go for production of your diploid nature of seed. Okay, without undergoing any sort of meiosis, right? But in case of apospory and adventitious embryony, there is the initiation of mitosis in vegetative cells leading to embryo or embryo sac development. In both the cases, as I told you, in apospory is the condition where embryo will develop from any cell
Cell, other
of the embryo sac other than egg cell, right? So any other cell in the embryo sac is undergoing development to form your seed. That is case of apospory. What happens in adventitious embryony? Yes, anybody? Adventitious.
Cells,
Hmm.
cells outside the ovule that are vegetative in nature, they form embryo.
Yeah. So basically,
embryo develop directly from
element.
vegetative cells of
Yeah.
Embryo sac is
Vegetatives.
not formed in adventitive embryony because
Embryo
that's
sac is not formed. Exactly. Embryo sac will not be formed directly. The embryo is formed from the vegetative cells of the outside the ovule, right?
ovule.
So in both the cases, what is happening is there is directly initiation of meiosis in the vegetative cells. Here there is some sort of modification of means that MMC is involved. But in these particular cases, the mitotic event is directly taking place in both aposporia and adventitious embryony. There is no role of your what? Meiosis. That's what they mean to say actually. Okay, so there is three basic components of apomictic pathway. First one is apomeiosis, that is the altered meiosis pathway in case seen in case of your apomixis. That's why it is called as apomeiosis. That is the production of that leads to production of unreduced megaspores. Second pathway is parthenogenesis, that is activation of egg cells to form embryo in the absence of fertilization. Means egg cell will be activated. The egg cell here also egg cell is present. This one is egg cell, here also egg cell is present. Here the egg cell is not excited to form the embryo until unless the it is getting fused with the male gamete. But here, because the parthenogenesis process is occurring, that process is making, means enabling this particular egg cell to form the embryo, right? That is the process of parthenogenesis. And another process is occurring in apomixis. You can see that is autonomous or pseudogamous activation of endosperm development. So autonomous development of endosperm means without excitation of your pollination, sometimes endosperm development takes place. That process is called as autonomous development of endosperm. Sometimes with the stimulus, after the stimulus of pollination is given, after that only endosperm development occurs. That process is called as, yes,
Pseudogamy.
pseudogamy. Yes. So these are the three typical peculiar feature which lead to your process of apomixis. So for the process of development of apomictic lines, there are not three, there are four basic. You can find the particular first three in several books. Chemical one I just found from one paper, but I'm not sure about its accuracy. I just found it, so that's why I put it here. I'll be doing little bits research regarding search regarding this one, chemical induction of apomixis, which particular chemical is involved. I'll tell you, okay? But I found it this from one journal. So we'll talk about this. So let's talk about the other processes. That is first one, that is gene transfer from wild species. So from wild species, as you know, they are the hub for the various genes, right? So from them directly doing the crossing, we can transfer the genes to cultivated one. So all the 4 points as I mentioned here, sorry, They are very important for your examination point of view. So anywhere you find any sort of source for anything like source for apomixis or source for cytoplasmic male sterility or source for any resistance, you should note that down and make a chart out of it, so that that will be beneficial, means easier for you for revision, right? So I found this four basically, so I put it here. If I'll be finding any extra information regarding this, I will tell you. So in case of your maize, the donor for the apomictic line is Trypsacum dactyloides. In case of your pearl millet, Penicetum, sorry, this spelling is wrong. This is M. Penicetum oriental and Penicetum squamulatum. Okay, Penicetum squamulatum are the source for the apomictic line in pearl millet. Then in case of your sugar beet, you can find beta trigyne and beta intermedia are the sources of apomixis. Then Elaima species is the source for transfer of apomixis to wheat. Okay, then we can do the induced mutations to create apomixis in the lines. We can use both physical and chemical mutation for this, and selection of apomictic genotype in the segregating generation. We can do that, means sometimes apomictic lines are spontaneously also created. So if you find any sort of abnormality in the plants, so there are some criteria looking at which we can tell that the plant may be undergoing apomixis process. So in the coming slide I'll be telling you how to detect it. So for detection of apomixis, this is for your knowledge. For JRF this is not that much important, but this is important for SRA purpose. So just for your knowledge sake, you know, uh, so if the when you can tell that there is a apomixis is present in the plant, if the condition is like, if one you have crossed the particular plant, means two parents, and you are not able to obtain the f1s from a cross, the condition might be that female is apomictic in nature, that's why you didn't get any seed when you cross the particular plants. Okay, and if there is a condition that there is uniform progeny in f2 population, f2 population's nature should be, it should be segregating in nature, but if you find uniform progeny in the f2 population, there might be a case, that might be a case of apomixis, right? Then uniform progeny from a heterozygous plant, from heterozygous plant or opal pollinated plant, if the progenies are coming uniform, so uniform means that will be similar to that of your maternal parent. So if that condition is there, means you can expect that apomixis is there. Then production of maternal phenotype in the progeny from a cross without selfing. If the selfing is not there and we have knowingly crossed the plant, still we are getting the plants like maternal genotype itself, sorry, maternal phenotype it is showing, that condition will be called as apomixis in the progeny. Then if the production of maternal progeny with recessive gene expression is there in the progeny, even if the male parent which we are pollinating, that is carrying a dominant thing, even in the presence of that dominant gene should express in progeny, no, but the recessive one is getting expressed, so we may indicate that there might be a condition of apomixis. Okay, then if you find multiple seedlings per seed, that condition might be because of your polyembryony, which is a case of apomixis. In the self generation, in the presence of plants showing no inbreeding depression, if the means there is no inbreeding depression is seen in case of your selfing, then you can guess that, means we can conclude that there might be a case of apomixis, because in self generation some sort of apomixis, some sort of inbreeding depression is bound to happen. Okay, whatever the case might be, some sort of inbreeding depression will be there, especially in case of your cross pollinated plant. This condition holds very true. Then embryological studies from the young ovules, if you will do, you can easily confirm the apomixis. So all these points which I have discussed over here is they are indicative of apomixis, but further cytological studies are required for confirmation of apomixis, right? So next thing is, what is the application of apomixis? So first ever application is fixation of heterosis. So what do you mean by fixation of heterosis? Is fixation means you will find this term used various times in various parts of plant breeding. So fixation means keeping it as such. Wherever you find fixation of a trait, fixation of a character, putting it as such means that will not
Allele,
segregate, that will not change any further. Whatever expression you are getting right now, that thing will be kept as such forever. That condition is
allele,
called, yeah, yeah.
allele frequency will remain one for such characters.
Allele frequency will remain
One for such characters. Can we say that?
one. Allele frequency is one. Are you telling that?
Yeah. In case
Uh,
of fixation.
no, allele frequency. See, there might be different alleles. No, anyhow, whatever the condition is, that total allelic frequency will be one only, right?
Yeah.
According to Hardy-Weinberg law, the total
Huh.
allele frequency will be one only. That varies that what is the exact. See, if there are two alleles, capital A, small a, so
Hmm.
initial the genotype means in the parental generation, let's say in F1, F1
Hmm.
the allele frequency is like zero point 0.4 and 0.3, 0.6.
Hmm.
So together they will form one only. In the
Hmm.
second generation, what might be the condition that because of selection or something else, this frequency may alter, but all total they will make one itself. Means whatever the condition, this condition might be like 0.2 or 0.8. This may be, if the Hardy-Weinberg equilibrium is not satisfied, means in
Hmm.
that condition the alteration is going to happen, but ultimately both these alleles are going to make together, the sum should be one any case. Okay, if Hardy-Weinberg equilibrium is maintained, means the zero point 0.4, zero point 0.6 will continue to remain like this. If it is not maintained, means you can see alterations in the allele frequency.
Point
That
two,
is the
point eight.
case. The
Hmm.
fixation of thing is that the allele frequency, yeah, you can tell that allele frequency will remain constant, means
Because
there
other
will
will
not
be
be,
variable.
yes.
Since other genes, sorry, other alleles are variable, so one can be set fixed if its frequency becomes one, so that it cannot be evolved more than that, or we cannot vary that particular allele.
One allele, one particular allele's frequency cannot be one anytime. That's what I wanted to tell you. Either
Ma'am, can
capital A or small a,
Hmm.
its own frequency will be less than one only. It can never be
Okay.
one.
can we say that total variability will be remain constant?
Yes, total variability will remain constant. Whatever variation is there, means in heterosis, in the heterosis that is in F1, you will find some sort of, means whatever find, means whatever variability is not there actually in F1, means all the plants are looking similar only. But whatever expression, whatever the combination of genes, whatever the allelic frequency is present in this particular condition in F1, that condition is going to be constant. Okay,
Hmm.
if it
Yes.
is not in F1, let's say if it is in F4, you are going to you had some plant that is in heterozygous condition, but there you have the apomixis condition, so you want to fix the particular line using the apomixis. So in that condition, because the plant is in heterozygous condition, so if you are maintaining the plant using apomixis, in that condition you can tell that variability is fixed or variability is kept constant, right?
Yes,
Yes,
ma'am.
ma'am.
Understood, no? Why I said variability is not there. Variability means the variation you can see within the genetic component or environmental component, total component of variation which you can see in the particular population. In F1, is there any difference between the plants? You can see any difference? No, no,
No, no, ma'am.
because all the plants are looking similar only, all the plants have same genotype only, right? So the plants are means plants have to be perform similar way. So there is no genetic variation present. If there is any sort of variation you can find, that is basically because of environment. So variability is almost zero in case of your F1. So we cannot say variability in case of F1. But if you are means selecting any plant, as we do in case of your sugarcane breeding, in case of sugarcane, what we do is we simply cross the plants. Okay, cross the plants means that is basically intervarietal crosses we do. So whichever plant is showing good uh, features, like it is having good cane yield or height or thickness or anything, that plant will be maintained constantly with the help of the asexual reproduction process. Okay, so in that way, in that particular condition, we can tell that the variability is fixed. The whatever heterosis the plant was showing, that was also fixed. Okay, if the
Okay.
condition present over here is pertaining to the population anything other than F1, we can tell that. But in F1 we cannot tell about variability.
Hmm.
Okay.
Yes, ma'am.
Yes, ma'am.
Okay, so how we are fixing the heterosis over here is, here we are crossing P1 and P2, two plants, two parents. So for any of the crosses, the minimum requirement that the two parents will be inbred in nature, means homozygous in nature, and they will be having extreme phenotypes. That is the basic requirement. Means as far as they possible, they should be distant in nature to have more amount of heterosis. So they will be giving rise to the F1. So whatever composition is F1 is having, in the first case only it gave rise to F1. After that it will be maintained apomictically. Okay, after that it will be maintained apomictically. So in this condition we can tell that the heterosis is particularly fixed. So no need to go for production of that F1s in all the generation. Means by the farmer can use their own seed for maintaining the plants. So this is the all the advantage of apomixis using this fixation
I
point.
mean, parents, there should be facultative apomixis.
Yeah, in that case facultative apomixis should be there. That is desirable. Okay.
Yes, ma'am.
My
मैम
throat is very
अगर
bad today. Yeah, bolo.
मैम अगर फैकल्टेटिव एपोमिक्सिस यहाँ पे हम करते हैं तो लाइन फिक्स रहेगा वो, मतलब आफ्टर जनरेशन सपोज 5 or 6 generations will it be fixed for longer duration?
ये slide एक बार So the apomixis leads to fixation of heterosis, since apomictic progeny of hybrids have same genotype as that of hybrid, right? Obligate apomixis results in complete fixation of heterosis. In such cases where apomixis is dominant, sexual reproducing lines is used as female parent, and obligate apomictic line is used as male parent to produce the hybrid progeny, that is apomictic. Okay, so how you will going for production of hybrid? So here the parent, female parent, which one you will use as female parent, that will be sexual reproducing one, and the apomictic one will be used as male parent. So if you are using obligate apomixis, means this will be leading to complete fixation of heterosis. As you said, if you are using facultative apomixis, that will give us a scope. Means that is not beneficial for farmer, but that will give the breeders a scope for, if someday something is happening to the variety, like it is getting susceptible to something or something like that, if it is a resistant variety, so we there is a scope of further development if it is facultative apomixis. But if you want the complete fixation, means we don't want to alter the condition in any of the case, we can go for obligate
Obligate.
apomixis. But obligate apomixis, if anything happens to the variety, means that variety will be completely lost. Means let's say heterosis will be fixed anyhow, but further development scope will not be there in case of your obligate apomixis. Okay,
It will
like
be
you
the
see
last
when we are,
generation.
yeah, it will be the last generation. Means we have to improvise the parents itself to do any sort of further development in particular obligate apomixis case. Okay, but
मैम
apomixis
ये
being,
जो
yeah,
मैम
बोलो।
ये जो आपने अभी बोला कि सपोज वो रजिस्ट सॉरी कुछ ससेप्टेबिलिटी आ गई या अगर वो वैरायटी लॉस हो रही है, तो अगेन वी कैन यूज दैट वैरायटी एज मेल पेरेंट एंड देन क्रॉस इट विद द फीमेल व्हिच इज ओपन पॉलिनेटेड और सिंपल फैकल्टेटिव
हाँ,
वन.
हम वो कर सकते हैं, वी कैन डू द क्रॉसिंग वर्क, बट this apomixis is means is a process where we can avoid, where the cross and making the cross is a difficult task. There they are telling that we can go for apomixis process, which will avoid making the crosses. In Hart's case we will do that only. We can, there is an option that
Yes,
we can change the parents and we can alter, we can use the hybrid itself as one of the parent and go for that. Okay, we can do that.
okay.
बट द थिंग इज दैट यहाँ पे हम लोग ना क्रॉसिंग नहीं करने का सोच रहे हैं, तो जहां तक हो सके हम क्रॉसिंग नहीं करके आगे बढ़ सकते हैं, उसको हम ज्यादा प्रेफरेंस देंगे। ठीक है, बाकी तो सारे ऑप्शंस हैं
Yes,
ही हमारे पास, जो भी पेरेंट्स, इनिशियल पेरेंट्स हमारे पास है, तो हम उसको इम्प्रोवाइज करके कर सकते हैं। राइट,
yes,
अंडरस्टूड?
yes, ma'am.
Okay, so what will happen over here is the parent, the female parent will be the sexually producing ones, so there will be no difficulty with production of seeds. Only the apomixis thing is getting transferred from the male parent into the progeny. So from here what will happen, the normal seed will be formed in the F1 seeds are normal, but it will have the apomixis, means whenever you are going for F1 generation, means when you are sowing the seeds of F1, that is the hybrid seeds are grown in the land, they will be giving rise to apomictic progeny. Okay,
Yes, ma'am.
seeds will be formed but they are apomictic in nature. So after this the farmer is using this particular seeds for selling, means this is the produce of the particular farmer is using this for selling. He can save this particular seed and sow in the next generation, because these are apomictic. They will in the next generation itself, that particular combination over here is that particular condition will be there itself, and they will grow seeds again in the next generation also. Okay, that is the benefit available with your what apomixis.
Apom.
Did you understand this what I said? Here F1 is there, no, F1 you got the heterosis, these particular plants are having heterosis. From here the seeds you got, seeds are sold by farmer, means this parental seeds you sold to farmer, and farmer has grown these seeds, and now the plants have come up, these plants are having apomictic seed. Farmer will now sell the particular produce in market, and he can save some part of his produce to grow in the next generation. No need to depend on the market or the particular company for getting the original hybrid seed, which is costly affair, right? So farmer can save some sort of amount of him, right? The hybrid will produce only apomictic seeds, which use for maintenance and multiplication and cultivation of the hybrid variety. So this particular condition is achieved in, it is noted term, actually spelling is mistake. TUM.
Ma'am, the real thing is that variability will not will be fixed. I said that it will be constant, but it will be fixed. Am
Yeah,
I right?
yeah. Fixed means the same thing, means same genotype you will find in all the generation, subsequent generation. That is the meaning of fixing.
Yes, ma'am.
Understood, no? Any
Yes, ma'am. Yes,
confusion?
ma'am. No, ma'am.
Okay. So as I told you, these are the two examples of your obligate apomixis. That is Penicillium notatum and Penicillium ciliary, that is the buffalo grass and bahia grass. These are the two examples of obligate apomixis. There they have did this program very successfully. Okay, as I showed you, like P1, P2, inbred line and inbred 2. This is the sexual one, this is the apomictic one, they crossed, and this is the F1 plant. From F1 they have raised the F2, and this is not typically, we cannot call this as F2 because here sexual reproduction is absent, but this is the progeny seed of F1, And after that also, in further generations also, the same sort of plant stature is maintained. That's what you can see over here. Next concept is that is hybrid. Hybrid is the progeny of cross between two facultative appomattox.
Somatic.
Yeah, yeah. Somatic hybrid is hybrid, not that is not
Hybrid. Yes,
that
ma'am.
is
I
not
was bit
hybrid. We will discuss about that in the next slide. It is there. So hybrid is the progeny of cross. We are crossing, physically crossing two apoplectic lines, facultative apomictics, and here its reproduction will be through, definitely through facultative apomixis only, and it is maintained via harvesting the seeds of F1 like, this is F1 like apomictic plants. Exactly F1s will not come because here apomixis is observed, means both the parents are apomictic, no. So in the original cross, what we are doing, one parent we are using the sexual one. That why it was easy. Normal seed set you will get. But here, because both the parents are apomictic, so normal one will not be there. So F1 kind of plant, whatever you will get, that will be used for maintenance of this particular hybrids. Okay. And these are expected to show constant and superior yields over generations. Means this will be somewhat like that of performance, that like that of your hybrids. Okay. So there are some other points which I want to discuss, like apomixis is a nuisance when the breeder desires to obtain sexual progeny. That if you want to sell the particular means if you want to go for sexual hybridization and if you find apomixis there, means it is a problem. So it is not of great help to in that particular condition. Okay. But for maintaining the variety, means once the desirable genotype you have selected, it could be further multiplied and maintained as apomictic progeny. But until the selection process is over, if apomixis is there, means it will cause a problem in selection and varietal development. Okay. So the advantages are like obligate apomixis permits the fixation of heterosis in the hybrid, as I told you. New variety or hybrid can be multiplied in a manner like pure lines. When simply you sow them and you just whatever seeds are coming up, you just take the good plants that are showing true to the performance to its parent, similar to any sort of self-pollinated crop. Like that you can just select the plant and further you can sell them or use for the next generation. Then here if the parents are having different time flowering time also, you can grow them to obtain very few hybrid seeds. Further you can just perpetuate those few hybrid seeds and in apomictic manner, because here hybridization is not a problem, means sexual hybridization is not involved, so no need to wait for the particular flowering time or season like that. Okay. And the nucleus seed of the hybrid varieties can be conveniently maintained. Why? Because here there is a very less chance of impurity, because whatever cross pollination or self pollination, whatever the condition is, here there will not be any sort of sexual progeny unless it is facultative type. If it is obligate type, means the seed multiplication is a very easy task. Okay. And here there is no need to maintain the isolation distance also. Pollen dispersal process is not at all relevant. Okay. Then it is a very complicated phenomenon, that is the biggest problem. And we don't have that much understanding of this apomixis. Then estimation of apomixis is a problem. It is not very clearly understood. Then sexual progeny sometimes means facultative apomixis will be affected by environmental factors, and genetic basis of apomixis is not clear. Okay. So that's all about apomixis. Just give me 5 minutes of time, I'll be right back. We'll start with um, self incompatibility. Okay. Hello. Okay. Let's begin with the next topic, that is self incompatibility. So there are two processes of fertilization, that is the promote the pollination or cross fertilization basically, or they promote the allogamy. The two important processes are like two important processes are like your self incompatibility and male sterility. Okay, these are two important processes. So both the processes will not lead to cell pollination or self fertilization. Can anyone tell me what is the basic difference, what is the major difference between male sterility and self incompatibility, based on your understanding? I just want to know.
In self-incompatibility, the pollen is fertile in nature, but in male sterility, it is not. Main reason.
Exactly. That is the basic difference. That in case of your male sterility, pollen is not at all fertile. Okay, pollen cannot fertilize on its own or any other flower. But in case of your self compatibility, pollen is not capable of fertilizing its own flower, but it is fully functional to fertilize the other flowers. Okay, that is the major difference between them, right? So so there are two fertilization basically. I wanted to show you. Okay. So the term self-incompatibility was coined by Stout in 1917, and first report of self-incompatibility was given by Colluter in Verbascum Phoenicum. This is very important. And what is the condition that will indicate you the self-incompatibility to be present in there? If there is a plant that does not set seed when pollinated with own functional pollen. Functional pollen is there, but still the plant is not setting the seed. In that condition, you can tell that self incompatibility is present. And the second condition is like, here the own pollen is not able to set seed, but it exhibits the normal seed setting when cross pollination is done. So the flower is responding to other pollens but not its own pollen. So that is clear cut indication of self incompatibility. Okay, so coming to the features of self incompatibility, it is like, first point I already told you, that is it does not set seed when self pollinated with its own pollen, but normal seed set is there. It is an outbreeding mechanism. Outbreeding means crossing. Okay, crossing, fertilization, which prevents autogamy. Autogamy means self-fertilization and promotes allogamy. It maintains high degree of heterozygosity. High degree of heterozygosity, why it will maintain means heterozygosity will be arising from the crossing. Cross fertilization, it will lead to development of heterozygous progeny, right? So this process leads to leads or maintains the amount of heterozygosity and reduces the amount of homozygosity. Homozygosity will increase when you are doing the self pollination or self fertilization. And the feature of self incompatibility can operate in any stage between pollination to fertilization. So the basically the stages which will come between pollination to fertilization are, the pollen has to, first of all, pollen will fall. Here the pollen will fall on the stigma, then the pollen tube has to germinate on the stigma. So the first thing where the barrier can be there, pollen and stigma, here the one interaction is there. Then the pollen tube has, okay, pollen has germinated on the stigma, it has to move through this pollen tube. So pollen grain and the style has to interact right now in pollen tube and style. So pollen style interaction is there. Then pollen tube has grown and it entered the ovule. Let's say it has grown this particular point and it has entered the ovule. So after entering the ovule, whether it will be able to fertilize the egg cell or not, so that depends on the interaction between pollen and the ovule. So in all the cases, there might be some sort of hindrance, or there might be some sort of what barrier might be there, which may affect your fertilization process. Okay, if all the processes are satisfied, if in all the cases there is no hindrances found, means the pollen tube will release the male gametes ultimately, and the double fertilization will be a success. Okay, So that is the point I wanted to explain over here. So what is the physiological effect of SI? That is how the SI actually operates. Means first of all, there will be failure to germinate on the stigma. Means the pollen grain cannot germinate on the stigma. That is the four stages I was telling you that I explained over here. If some pollen grains do germinate on the stigma, the pollen tube will fail to enter the stigma. Means the stigma surface will have some sort of stigma surface actually provides a hydration to the pollen. After that only pollen from the pollen tube, from the pollen the pollen tube can originate. Okay, so if that sort of hydration is absent, means pollen grains may germinate, but for the pollen tube growth will be absent. And if there is any sort of callose deposition, sometimes we can find in below stigma surface. So pollen grains will germinate, but the pollen tubes cannot enter through those callose depositions. Those sort of thing you can find in case of your sporophytic self incompatibility, basically. That is in case of your brassicaceae family. Rye, radish, cabbage, all these things you can find there. If the pollen tube enters the style, but they grow too slowly to affect the fertilization before the flower drops. Means, okay, third case might be like the pollen tube entering the style normally, but the pollen tube growth is very slow. By the time it reaching the ovary, the flower is already, means its viability is gone, flower has already dropped, so fertilization is not possible. Other condition is like when sometimes fertilization is affected but the embryo degenerate at very early stage. That condition will be called as late acting self incompatibility. Okay, so there all the steps have been normal, means up to the fertilization process, even the fertilization is there, but the embryo is not able to survive. So it will degenerate in the very early stage. That condition is called as late acting self incompatibility. That is found in sugar beet, in cocoa plant also it is found. There are two hypotheses regarding the self incompatibility, whether there will be stimulation or there will be inhibition. Okay, so based on that there are two type of hypotheses. So the complementary hypothesis states that it was proposed by Batman 1952. So it tells that in case of your like genotypes, there will be absence of stimulation or pollen germination. That's why there is self-incompatibility is resulting in case of your complementary hypothesis. And in case of your unlike genotypes, there will be ample amount of stimulation present. Stimulus will be there for pollen germination, and there is no incompatibility reaction found in that. So like genotype means here you can see this is the genotype of the one parent, that is S1 S2, and this is the genotype of other parent, that is S1 S2. Both are similar, that's why there is absence of stimulation for pollen germination. That's why the uh, incompatibility is occurring. But here you can see S1 S2 is there in one parent and S3 S4 allele is there in another parent. That's why you can see there is no incompatibility and success of pollen germination is there. Oppositional hypothesis states that the interaction between the alleles, that is same alleles, leads to production of an inhibitor. There will be inhibitor produced, which is produced because of the interaction of similar sort of allele, which inhibits the growth of pollen tube in the pistil. If there is unlike genotypes are there like S1 S2 and S3 S4, in that condition this inhibitor will not be produced, so the pollen tube growth is normal. So these are two hypotheses only, means based on that different scientists have proposed. Like there might be some sort of stimulus chemical might be acting, or there might be some sort of opposition might be acting. Okay, up to this point is it clear?
Yes, ma'am.
Hello, is it clear, students?
Yes, ma'am. Yes, ma'am.
Okay, so let's move to the next thing, that is classification of self incompatibility. The classification was given by Lewis in 1954. This is important to remember who was given the classification. That is Lewis in 1954. So he has given the incompatibility reaction based on, first of all, whether the difference means the incompatibility mechanism is because of flower morphological difference or not. Based on that, there are two systems, that is first one is heteromorphic system, second one is homomorphic system. Here there is the morphology of here the flower morphology itself causes the self incompatibility. Okay, and this self incompatibility is not because of gene, and not because of physiological effect. I'm sorry. So here basically two types of thing you can see, that is distyly and tristyly. Okay, let's move on to heteromorphic system. Here the SI is associated with flower morphology, as I told you. So distyly refers to a condition where you can find two types of style. Okay, two types of style, that is short and long. Means style is short and long, and stamens low and high. Let's see what is the condition is there. So here you can see two types of flower. This is the flower of the particular plant, that is primula. So here you can see this is the style, this is the anther. Okay, so in pin flower you can see the central one, that is the stigma is up, means it is taller and the anthers are in the lower position. Thumb flower condition, there is the opposite condition where anthers are placed in the top and the style is shorter. So pin you just imagine like this you imagine the pin. In the central part of flower, in central part of flower, always and always stigma is there, right? So in case of a pin flower you can see, sorry, stigma is taller one. Okay, you just remember pin flower, thumb flower you will remember easily. Pin flower, this is somewhat conical and sharp towards the center. So here stigma is, means style is taller and the anthers are in the downward position. Okay, this is the pin flower, this is the thumb flower.
मैम आज क्लास यहीं से बंद कर दीजिए, आपका तबीयत सही नहीं है।
कोई
हाँ,
बात नहीं, मुझे ही
नहीं
लेना
मैम,
पड़ेगा ना थोड़ी।
नहीं नहीं मैम, कोई बात नहीं मैम, आप नेस्टलस ले लेंगे, कोई दिक्कत नहीं मैम। मैम
देख,
मैम
बस
छोड़ दीजिए।
हेट्रोमॉर्फिक खत्म कर देते हैं, फिर क्लोज कर दूंगी। हाँ, एक्चुअली मुझे भी तबीयत इतना सही
नहीं
नहीं लग रहा है।
मैम, मैम नेस्टलस पढ़ा लेंगे, कोई दिक्कत नहीं मैम। नहीं तो आप कहीं कभी फ्री हो गए तो आधा घंटा 1 घंटा ले लेना, अभी टेस्ट कर लीजिए मैम, सही है।
थैंक यू सो मच फॉर केयर। ओके, हेट्रोमॉर्फिक खत्म कर लेते हैं, ठीक है, 5 मिनट में। चलो, तो देखो यहाँ पे पिन थ्रम फ्लावर हो गया है ना। उसके बाद दिस वन इज पिन फ्लावर वेर यू कैन फाइंड द लॉन्ग स्टाइल एंड शॉर्ट स्टैमन, and thumb flower you can find short style and long stamen. Okay, here the only condition that is compatible mating is pin and thumb flower. Means we can only cross the pin and thumb, then only we can get the successful f1s. Otherwise pin and pin if you will cross, means you will not get anything. Thumb and thumb you will cross, means you will not get anything. Okay, and here the examples are important, that is sweet potato, and in buckwheat you can find this sort of distyly condition. Okay, this is important, and basically we can find in other plants of primulaceae. Tristyly means here you can find three types of styles, means anther and styles have three different positions, that is short, medium and long. And the example is important, that is lythraceae. Okay, this is the example of Lythrum salicaria. Here you can find the short style is, so this one is style. Okay, this is short, medium and long kinds of stamens you can see over here. Style is medium, here is the style, this one is style, it is medium, and stamens are short. This one is stamen and this one is stamen, short and long. And this one also you can see the yellow arrow here, the style is long but the stamens are short and medium. So in any of this condition the mating is not possible with similar kind of flowers. Only and only opposite kind of flowers, means this type of flower can, this type of flower can mate with this one, this type of flower can mate with this one. Among them we can cross, but own flower cannot fertilize itself. Okay, that is the condition of tristyly. Okay. Automorphic system. Okay. Any doubt, student, till now
No, ma'am.
in apomixis or here?
No, ma'am.
Okay. So thank you so much.
So much.
My health is
My
not that
health.
good actually. I'm just sneezing a lot, and I got a little bit cold because of continuous rain and all. Okay, so I'll be stopping here right now. We'll be taking some other day during the weekdays. If I'll find any time, I'll take the class. And for economic botany, I hope means it is not proper. Means right now I can understand the way I'm going to the economic botany section. It is not proper. Means you are not finding the right way. From the next session, I'll be telling you which exact position portion I'll be discussing from economic botany also, so that you can have a better idea. Means you study little bit before coming to class, so that we can have a good discussion, right? Okay, thank you all for
Thank
your
you.
patient hearing. Okay, and then I'm going to close the session. Bye bye, good night.